Welcome to zoology class. So in the last class, we have discussed the structure, the molecular structure of the DNA molecule proposed by James Watson and Francis Crick. Now in this class, we shall discuss about the packaging of a DNA molecule in a cell. Packaging of DNA molecule in a cell. And you may wonder, what is the term packaging is related with the DNA molecule? Now what do we mean packaging? Packaging is the way of managing something big, something large in size, for better transport or for better handling or for conveniently placing it somewhere. So for example, we package our dresses for a journey into a suitcase or into a bag. We cannot carry our dresses like shirt, trousers, sari or whatever may be for a journey as it is. You know that it is very inconvenient and it is not possible. So in order to conveniently manage our dresses, we pack it to a convenient size and place it in a suitcase or bag so that we can carry it, transport it in a convenient manner. So in the same way, DNA also need to be packaged. Of course, because DNA is a lengthy molecule. How long a DNA molecule is? Let us calculate the length of a DNA molecule in human being. So in the last class we had seen that the length of the DNA molecule depends upon the number of nucleotides present in the DNA molecule. So in human genome, the length of the DNA molecule, the number of nucleotides in the human cell, number of nucleotides in a DNA molecule in our cell is 6.6 into 10 to the power of 9. That many nucleotides are present in a diploid condition in our cell. That is 6.6 into 10 to the power of 9 nucleotides are present in the DNA molecule in a diploid condition. And the distance between two nucleotides in a DNA molecule is 0.34 nanometer. It is 0.34 nanometer. And 1 nanometer is equal to 10 to the power of minus 9 meter. So when we convert nanometer to meter, 1 nanometer is equal to 10 to the power of minus 9 meter. So 0.34 nanometer is equal to 0.34 into 10 to the power of minus 9 meters. When we convert the length of the DNA molecule into meters, length of the molecule is equal to the number of nucleotides in a DNA molecule, and the number of nucleotides is 6.6 into 10 to the power of 9. And when we convert this length of DNA molecule into meters, that becomes 6.6 into 10 to the power of 9. That is the number of nucleotides multiplied by length in meters between the two nucleotides, that is 0.34 into 10 to the power of minus 9. So that is 6.6 into 10 to the power of 9 into 0.34 into 10 to the power of minus 9. So that is 6.6 into 0.34. That is equal to 2.2 meters. Astonishing, is it not? So the DNA molecule, the length of the DNA molecule when it is converted into meters, it is 2.2 meters. So 2.2 meter long DNA molecules we have in our cell. And a cell is such a minute unit of body. And the DNA is situated in the nucleus, which is still smaller. And the size of the molecule, size of the nucleus is calculated to be 10 raised to or 10 to the power of minus 6 meters. So 2.2 meters of DNA should be situated in a small space such as the nucleus, that is only 10 the power of minus 6 meters. So, in order to contain such a long DNA molecule in such a tiny space like nucleus, it should be packed properly. So DNA need to be packaged or packed properly in order to contain it in a nucleus which is very small. The size of the nucleus is very small compared to the length of the DNA molecule. And the packaging of the DNA molecule is different in prokaryotes and eukaryotes. What's a prokaryote? A prokaryote is an organism whose cells are without a well-defined nucleus. A proper nucleus is absent there. Nuclear membrane, nucleoids, etc. are absent there. And DNA are let loose in the cytoplasm. So that doesn't mean that DNA are spread all over the cytoplasm. There is some packaging there also. In prokaryotes also, some packaging is there. So let us see how DNA is packed in a prokaryotic cell. So in prokaryotes, DNA becomes helical loops, as you can see in the diagram. It becomes helical loops held together by means of positively charged histone proteins and non-histone proteins. These are proteins. So positively charged, DNA is negatively charged. Due to the presence of phosphate group, DNA is negatively charged. And in prokaryotes, this DNA becomes helical coils. DNA itself is helically coiled, ladder-like structure we have already seen. But a long DNA molecule becomes helical coils, as you can see in the picture here. And these coils are held together by means of positively charged histone and non-histone proteins. And this structure is called a nucleoid. Helical loops of DNA molecule held together by histone and non-histone proteins is called a nucleoid. Such several nucleoids can be seen, and DNA is situated in the cytoplasm of a prokaryotic cell in the form of these nucleoids. So several such nucleoids can be seen. And even the helical loops, each helical loop may again be supercoiled in some cases to reduce the length of the DNA further. Okay, so this is the way, in the form of nucleoids, in prokaryotes, DNA is packed and managed in the cytoplasm. In eukaryotes, the packaging of the DNA is in a different manner. The packaging, in the process of packaging, histone proteins play a very important role. Histone proteins, positively charged histone proteins play a major role in the packaging of the DNA molecule. In eukaryotes, the packaging should be more efficient, because the DNA molecule is contained in a nucleus. So, it should be packaged, the DNA should be packaged in a more efficient manner. So, for this, histone proteins and to some extent certain non-histone proteins also help. And these histone proteins are of different types. Different types of histone proteins are there. H1 histone proteins are there. H2A, H2B, H3 and H4 are different histone proteins. H1, H2A, H2B, H3 and H4 are the different types of histone proteins involved in the packaging of the DNA molecule. Here, two H2A histone molecules, two H2B histone molecules, two H3 molecules and two H4 histone protein molecules, totally 8 histone molecules, two H2A molecules, two H2B molecules, two H3 molecules and two H4 histone molecules, totally 8 histone units. 8 histone units combine to form a histone octamer. This is called the core histone protein. That forms a core, a spherical core, histone octamer. And DNA molecule gets wrapped around the histone octamer. The DNA molecule gets wrapped around the histone octamer. So over a single histone octamer, histone core, 200 base pair length of DNA molecule is wrapped. So a single histone octamer has a wrapping of DNA molecule to the length of 200 base pairs. And after this wrapping of 200 base pairs around a histone octamer, the DNA molecule comes out of this octamer, runs a short distance, then it again gets wrapped around another octamer, histone octamer, to a length of 200 base pairs. So it goes on like this until the complete DNA molecule, until the entire length of the DNA molecule is completed. So this unit, so this structure appears like beads on a string. It appears like beads on a string. And this is called a chromonema. Such several chromonemata can be seen in the nucleus. So in this chromonema, the beads are, each bead is a histone octamer around which some length of the DNA molecule to the extent of 200 base pairs is wrapped around. That is one bead. And this bead is called a nucleosome. This is termed as nucleosome. And this DNA molecule, the wrapping of the DNA molecule around the histone octamer is strengthened or supported by H1 histone. So H1 histone is not a part of the histone octamer. And this H1 histone is seen outside this histone octamer to bind together the wrapped DNA molecule around the histone octamer. That you can see in the diagram here. So, this is how the DNA is packaged. So, when this is done, the length of the DNA molecule can be managed to a considerable extent. So, it can be contained in a small space like nucleus. So, instead of letting the DNA molecule lay as a single long molecule, this DNA molecule is wrapped around the histone octamers at intervals. So on each histone octamer, the length of the DNA molecule wrapped is to the length of 200 base pairs. Then after the binding, after the wrapping of 200 base pairs around a histone octamer, the DNA molecule comes out free for a short distance, for a short length. Then again, it gets wrapped around another histone octamer for 200 base pairs. After that, it comes outside, and then it gets wrapped around another nucleosome, so another histone octamer for again 200 base pairs. So it goes on like this till the end of the DNA molecule. So that structure of histone octamer plus the wrap around DNA together constitute a structure called nucleosome. A structure called nucleosome. Okay. And this is how we see the chromatin fibers or chromatin threads in an interphase cell. During prophase until metaphase, there is chromonema, chromatin threads become short and thick. So this process of becoming the chromosome, the chromatin fibers short and thick is initiated in the prophase and continues till the metaphase, and it becomes, when it reaches the metaphase stage, the structure becomes short and thick, then it is called the chromosome, you know that. So during the metaphase stage, when the cell passes from the prophase stage to the metaphase stage, the DNA molecule coils, the coiling of the DNA molecule increases further so that the chromosome, the DNA becomes more short and thick. Then we say the structure chromosome. So chromosome is made up of protein and DNA molecule. This is how proteins are involved, or proteins are present in the chromosome. So proteins of the chromosome help the DNA molecule to get wrapped around and package it in a proper manner. And in such cases, in higher coiling, when a higher coiling of the DNA molecule takes place, non-histone proteins, non-histone chromosome proteins are also involved. So at the beginning of the package, the initial stage of the packaging of the DNA, histone proteins are involved. And when the DNA molecule undergoes higher coiling outside the nucleosome to make it more short and thick, then non-histone proteins are also involved to bind the coils together, to hold the coils together. Okay, so this is the mechanism of packaging of DNA in an eukaryotic nucleus. And when DNA are stained by using certain stains, by using certain dyes, it was observed that certain region of the chromatin are darkly stained and certain regions are lightly stained. The darkly stained regions of the chromatin are called heterochromatin, and the lightly stained regions are called euchromatin. So two regions can be identified in a chromatin fiber, in a chromatin thread. When stained by using certain dyes, certain stains, certain regions of the DNA or certain regions of the chromatin appear darkly stained, they are called heterochromatin, and certain region appear lightly stained and they are called euchromatin. And heterochromatin are considered to be non-functional as far as genetic gene expression is concerned. Darkly stained heterochromatin is considered to have less role in the gene expression. What do you mean gene expression? Gene expression means the expression of a phenotype, expression of a character by a gene. You know that genes are responsible for the phenotypic expression. Phenotypic expressions or phenotype, different types of characters, different character features are produced by genes. So that is called gene expression. So for a gene expression, for a gene to express in the form of phenotypic characters, heterochromatin has less role, or it has no role. Genetically, heterochromatin is considered to be inactive, whereas euchromatin is genetically active and has a major role in the gene expression. Okay, so we shall wind up the class here.